You'll remember that earlier we mentioned the fact that nervous tissue consists of two types of cells, neurons and neuroglia. Neuroglia is also referred to as glia. In the central nervous system, these are the types of cells that are considered neuroglia: astrocytes, oligodendrocytes, microglia, and Ependymal cells. We will focus on oligodendrocytes and Ependymal cells. In the peripheral nervous system, there are Schwann cells and satellite cells, and we'll focus on the Schwann cells. Let's take a look first at oligodendrocytes. You can see an oligodendrocyte shown here. As we can see in this diagram, each oligodendrocyte forms a myelin sheath around more than one axon in the central nervous system. We'll talk more about these myelin sheaths, but as you can see, they are wrappings that are found around some axons. Oligodendrocytes are analogous to Schwann cells in the peripheral nervous system, and what this means is that Schwann cells likewise can produce myelin sheaths around axons. Another type of neuroglial cell found in the central nervous system are Ependymal cells. In this diagram, we can see in the brain this blue area. These are cavities that are known as ventricles, and they're filled with a fluid known as cerebral spinal fluid. This fluid is also found in a portion of the spinal cord shown here called the central canal. Within the ventricles, these Ependymal cells, in conjunction with specialized capillaries, produce the cerebral spinal fluid. This diagram shows a little more detail concerning the cerebral spinal fluid. As we can see here, there's something called a choroid plexus that's found in this location as well as other locations. And these choroid plexuses, as mentioned before, consist of capillaries and Ependymal cells, and they are what's producing the cerebral spinal fluid, which is found circulating throughout the outside of the brain and also, as these arrows indicate, around the spinal cord and through the central portion of the spinal cord called the central canal. Schwann cells are the cells of the peripheral nervous system that produce myelin sheaths. This diagram shows the myelin sheath, and we'll look at this in more detail later in a special section on myelination. Each cell produces part of the myelin sheath surrounding an axon in the peripheral nervous system. So in other words, this is one Schwann cell producing a myelin sheath. This is another Schwann cell producing the myelin sheath of an axon in the peripheral nervous system. Some Schwann cells can surround unmyelinated axons as well. Let's now take a look at myelination. Myelination begins during the fetal development. In this series of pictures, we're taking a look at a Schwann cell and how it forms a myelin sheath. As you can see, the cell grows in a spiral type of fashion. Eventually, it results in what is called the myelin sheath, and then the outer portion, which consists of the cytoplasm and the nucleus, is called the neurolemma. This diagram also points out that a single Schwann cell can enclose as many as 20 or more unmyelinated axons, that is, axons that lack a myelin sheath. Another feature that we can see that is characteristic of myelinated axons is the fact that the myelinated sheath is not continuous down the length of the axon. Rather, there are gaps in the myelin sheaths, and these are called nodes of Ranvier. We'll look at the significance of these later when we talk about the transmission of an action potential down an axon. Let's now take a look at myelination produced by oligodendrocytes. As we can see in this picture that we've seen before, each oligodendrocyte myelinates parts of several axons. And in the case of myelin sheaths produced by oligodendrocytes, there is no neurolemma, the outer layer, because the cell body and nucleus of the oligodendrocyte do not envelop the axon. So these myelinated sheaths, in summary, are produced by oligodendrocytes in the central nervous system and the Schwann cells in the peripheral nervous system. The purpose of myelin sheaths is to insulate the axons and to increase the speed of nerve impulse conduction down the length of the axon.